Thank you so much. You've been so supportive since I've come into the Zenzino family, and you know me, I like to do a lot of digging before I decide to do something. So for me, I'm a clinician. I've been in the wellness industry for 20 years, and I used to run this test in my own clinical practice. I used to send it over to the US, and it was much more expensive, and I had to wait 6 weeks to get the test results back. But I absolutely loved what it told us, because we're dealing with inflammation, we're dealing with key raw building block nutrients for the body, and particularly for the nervous system. You know, nourishing the nervous system is one of the key things that we can do these days. So for me, when I was doing the digging, I wanted to be sure that the laboratory was gold standard, because as a naturopath, a natural medicine doctor, we have access to a lot of really cool functional medicine tests. However, they are very expensive, and so it's a fine line between getting people to invest in the testing and getting people to invest in the treatment, right, and my services. So it would become very expensive quickly. And this is when I was introduced to this concept of Zenzino, where they had the test that I used to do in a neat little protocol where there was a baseline test. We did some food-based nutritional intervention, and then we retested. And for me, a lot of the time, my clients would run out of money to do the retest, and they weren't that interested. But I was, of course, because I wanted to see if my therapy had been effective with them. So when I came across this little protocol, I got super, super excited. The fact that it was very affordable and it was gold standard. So I started looking into the laboratory and the people that run the laboratory, and I got super excited, and I feel very, very privileged to be talking to and working with Thomas Gundersen tonight. So Thomas shared with me that he's got some incredible information to share for everyone. I have actually seen some of this information, but it is really amazing stuff. We need to know this, because if you understand how the test works, you will understand the importance of this test and why it is such an incredible protocol to run with people. So Thomas, thank you so much for coming on and joining us here in Australia. We're looking forward to it. So I'll hand it over to you to share your slides. And then towards the end, we might have a few questions. If you haven't managed to answer everything that my little mind likes to go and explore, then I'll ask at the end. So thank you.
Sure. Thank you, Cherie, and thank you for having me on this Zoom conference. I will share my screen then. Yes?
Yes.
Okay. So this presentation is a version of a presentation I hold different places in the world, including Stockholm, in Globen, at the annual event a couple of times. So it's about the fatty acid profiling in dry blood spots, but it also covers a few slides about myself, the history of the company, what we do for other companies around the world, and especially then what we do for Zenzino. So this is the one slide about myself. I'm a doctor. I have a PhD in analytical chemistry from the medical faculty at the University of Oslo. I was the founder of this company 26 years ago. And at the same time, for 16 years, I also was a researcher at the University of Oslo. So in addition to my master, I have the PhD. I also have about 50 scientific publications, and I've been the PI in 6 EU-funded projects. So my company, Vitas, was established in 1994. And as you learn now, it's a spin-off from the University of Oslo, the medical faculty. Since I was running this lab there for 16 years, it's kind of an academic lab going commercial. So it's kind of different. It's not like a startup going commercial right away. It's an academic lab having the kind of the academic thinking and then going commercial. So our main customers are big pharma companies, biotech companies, academic institutions, food and nutrient producers, and hospitals, in addition to a number of startups within personal nutrition, precision medicine, and so on around the world. So we have a GMP certification, This is a quality certification you need to work with human clinical studies and also to release drug products to the market. So we are located in this building. This is the Science Park in Oslo. It's a building, it's built over 25 years. It's more than 300 companies, 3,000 people working with some sort of idea they have at the university and trying to crack the code, raise a business, or do something good for society. So we are at the top level on this building, and actually we have the entire floor here now. It's soon 1,000 square meters. So I usually say that we are 58 workers in this company now, where 23 currently are humans and 35 are robots or machines. And this is actually an important strategy of this company to employ 2 robotics systems for each human. And as you know, this is the modern way of doing production. So these robots, they work all night, helping the humans feeding the robotic systems. So this is how the robots really look like. It's not like the kind of fairy tale type of robots. It's kind of boring. They are quite expensive, ranging from 40,000 euros to 400,000 euros per piece. We, as a spin-off from the university, are also quite research intensive. So this is a list of some of the EU-funded projects we have been partners in. So this is from the 7th framework program. You can see institutions like University of Cambridge, TNO in Holland, University College Dublin, and some institutions in Holland and Belgium. Currently, we are a part of this project called LifeBrain. So it's a Horizon 2020 funded project, 10 million euros, 5 years to study the human brain from birth to death, with emphasis on dementia, Alzheimer, and depression. So in this study, MRI scans of the brain, about 4,000 people from all over Europe, are combined with blood biomarkers developed on dry blood spots by Vitas. So in addition to the academic project, we also have a number of customers. So this is a list of logos from some of the companies that we've been doing work for the last 6 months. So you can see it's a mix of academic institutions, big pharma companies, and smaller biotech companies. Our biggest customer, and I would say most important customer, is Zinzino. So this company has been with us for about 7 years now, and it's been growing quite impressively the last 2 years. I guess you've seen that the stock price has raised quite dramatically the last 2 days, which is probably related to the launch in Asia, I guess. And for this company, we do fatty acid analysis. So the classical way to do fatty acid analysis, or to do biomarker analysis, is to take a blood sample from the arm. So you've probably all done this at the doctor's. It's a big needle stuck into your arm, and you draw like 5 to 10 milliliters in multiple tubes. And when this blood is centrifuged, you will have a red layer at the bottom and a yellow layer at the top. The yellow layer is the blood plasma, and the red layer is the red blood cells. So this Zinzino kit has a totally different solution. In this case, it's no big needles. It's just a tiny drop of blood collected from the fingertip. And when this drop of blood is dried on the paper, it's called dried blood spots. And this is how to do it. You've probably done it many times yourself. You prick your finger, you drip the blood on the paper, you try to fill both circles. And this is a video. I can see if this is going to work. This is me sampling in a what I would say near perfect way. So you put the paper on the table, you prick the lower side of the longer finger, you squeeze gently, and you will have a large drop hanging from your finger. You should be patient until the drop is really large, and then you try to fill one circle with one drop. If you're not able to complete the whole circle with one drop, just add another drop right after. Then the second circle. So as I told you, you have the red blood cells at the bottom and you have the yellow blood plasma at the top. These red blood cells, they are born in the bone marrow. And actually every second, 2 million cells are born, and at the same time, 2 million cells die. So 2 million in, 2 million out. And when they are born, they live for 120 days. So when you take a dose of the balanced oil, the blood concentration of EPA and DHA will reach a maximum about after 6 hours. And when you take the second dose, you will have another peak after another 6 hours, and so on, and so on, and so on. So this blood is then going into your heart, and your heart will pump this blood throughout your body to the brain. And actually, DHA is really important for the brain. Actually, about 60% of the lipids in the brain is DHA. So what we really want to make sure is that we have sufficient amounts of DHA in the brain. So the best way will then to take a sample from the brain, right? But we cannot do that. So instead, we are looking at the red blood cells. And these cells are kind of tissue. They are cells floating around in your veins, and they are easily accessible by taking a small blood samples. So by studying these red blood cells, we can say something about other organs in your body, like the brain, your heart, and so on. And this is why we refer to these cells as satellite tissue. So the blood from your heart goes into your brain and your bones, and in your bones, the red blood cells are born. So when you have this increased concentration of omega-3 going into the bone, you will have newly born red blood cells with high omega-3. And at the same time, old cells with low omega-3 will die. So over time, you will have more and more red blood cells with high omega-3. And in this way, the concentration of omega-3 in your blood cells will increase. This is the fat blue line here. So in the blood plasma, the yellow fraction, you will only have this up and down kind of pulsing from your daily dose of omega-3. But over time, the concentration will not increase substantially. But so this is the blood plasma and the red blood cells separated. But if you put this on top of each other, which is the case in the dry blood spots, it will look like this. So this is the increasing background in the red blood cells. And in the beginning, before your first dose, you will have a low concentration. And then the spiking might be quite evident on top of the background from the red blood cells. But after some 30, 40 days, the concentration in the blood cells will be so high that the spiking will almost not be visible at all on top of this background. So Cherie, do you have any questions to this? Because now I'm going to switch to the technique that we use for analysis.
No, that's really good. I think you've covered off on a lot of things there, and I will wait until you've gone through the technique, because the couple of questions I have might come up in that. Yeah.
Okay.
Thank you.
Cool. So the technique that we use to measure these fatty acids in the blood samples is called chromatography. And chromatography is a Greek word. It's a combination of chroma, which means color, and graphy, that means writing. So it means writing in colors. And the reason for this is that it was originally used to separate the colors of the leaves in the autumn. As you know, when the green color disappears into the roots of the trees, the green, the yellow, the red, the yellow, and the orange is visible. And these carotenoids, they were separated by chromatography 120 years ago. So we use it for separation of fatty acids, and this is the result from such an analysis. This is what we call a chromatogram. And you can see some blue peaks here. You can see this blue high peak is arachidonic acid, the omega-6 fatty acid. And then you have the EPA, this small peak, which is the omega-3 fatty acid. And these two goes into this ratio, the omega-6-3 ratio. And you can see that this peak is about 5 times higher than this EPA peak. So in this case, the ratio is 5:1. So if you look at this ratio for 6 different samples, you can see AB, EF up here. So AB has a lot of blood and EF has little blood. So when we have a lot of blood, we have high peaks. And when we have little blood, we have small peaks. But if you look at the ratio, it's always 3:1. So this is a very nice feature for this analysis, and it's very well suited for home collection because we don't always have perfect samples. We have very different samples. So you can see now that it doesn't really matter how much blood you send, the result will be accurate anyway. And this also goes into the dilution that we discussed, Cherie. So some people say that if you squeeze your finger a lot, you will have cell liquid going into the blood and the blood will be diluted. And this is correct. But and for most analysis, this will be not good. But since we are measuring the ratio in this case, it doesn't really matter because both peaks will be diluted and the ratio will stay the same. So in this slide, I have really tried to provoke the sampling. You can see I have all different kind of samples. We have excess blood. We have multiple drops on top of each other. We even have one card with no blood. And these blue bars, they represent the result for DHA, one of the omega-3 fatty acids. And you can see no matter what we do with the samples, we still have approximately the same result. Except for this card with no blood, okay? No blood, no result. Makes sense. But look at this slide. So on this card with no blood, we actually have higher results than we have when we do have blood. And this is not DHA, this is C16, a very common saturated fatty acid. You will find this fatty acid in any type of lipid like butter, hand cream, and so on. So what do you think happened here? Why do we have higher results with no blood than when we have blood? So in this case, I touched the paper with my fingers, okay? And the reason we have so high results is that the lipids we have on our fingers naturally or by hand cream is much, much higher than what we have in the blood. So just a slight touch on the paper with your fingers will actually affect the results quite dramatically. So don't use hand cream and make sure to wash your hands with soap, warm water, and rinse well and dry off with paper before you do the sampling. So then a question that we often get is, I mean, Oslo is so far away from Australia, Florida, and Hong Kong, and so on. Does it really make sense to have a lab in Oslo handling all the samples from all over the world? So from the Nordic country and Western Europe, we have envelopes coming in by the normal postage systems, and they have stamps on. And stamps are quite rare these days, so it's kind of exciting to see these colorful, beautiful stamps from all over the world. And since we get something between 1,000 and 2,000 envelopes every day now, it's quite substantial. So we donate these stamps to stamp collectors through an organization called Tubfrim. So last year we donated a stamp for 150,000 Norwegian kroners. That's about 15,000 euros. And this is to the benefit on research or tuberculosis. So this year we think it will be about 30,000 euros, and maybe next year 40,000 euros, and so on. And soon we will donate something like 100,000 euros to research every year, or you will donate. So this is a good thing. So from more remote locations, we use air transport, and we have air transport currently from Florida 3 times per week, and I think from Brisbane or Adelaide in Australia, I think it's now about 5 times per week. And we are looking at Airbridge from South Africa as well, and Moscow. And I think in the future, we will have multiple air hubs around the world flying Cincino samples to Oslo for analysis. And it's quite fascinating because UPS, the transporter that we use, they guarantee less than 48 hours from anywhere in the world to Oslo. And the cost is something about 20 to 40 euros for a package of a couple of hundred samples. So this is a very good solution, working very well, and I think this is the solution for the future.
Thomas, can I just stop you there for a minute, because we've actually had a question come in from Petra. She said, tests from Germany, Switzerland last 4 to 8 weeks to get evaluated. Could you so I think, Petra, what you're asking is it's taken about 4 to 8 weeks to get evaluated. I know that here in Australia, Rob actually has set up a system whereby there's an extra envelope and we send them all to Rob, and then he express posts them over in a big package to you, Thomas. I'm wondering whether, Petra, maybe, I'm not really sure what's going on with COVID and all of that sort of stuff, but have you known, Thomas, why that might be the case, or why there's such a delay?
Yes, I think it's related to COVID to some extent. Usually it's only a few days from Germany, actually, to Oslo. And the average time for processing the sample in the lab is 3 to 4 days from arrival. So it's definitely the posted system in the local countries. And we have seen some big delays due to COVID since March.
And can you actually tell us then, how do you order what comes in? Like, because I guess they're not dated, right? So is there a system whereby you're processing each day the ones that come in? Yes?
Yeah. I mean, the samples that come in first, they are handled first. So it's a maximum of 7 days from arrival. And on average, it's 3 to 4 days.
Okay.
So,
and Cherie, that's why from our side we see such quick service, because like in Australia, I've trained everybody to try to use Express Post. So the quicker it hits my desk on a Wednesday and a Friday, the quicker it hits over to Oslo.
Sure, and that actually brings me to another question then, Thomas. You've covered off most of the things that I wanted to ask, but could you just talk a little bit about how dry does the blood have to be? If somebody accidentally puts it in wet, they don't know that step. Would that do something? And because there's a preservative in there, right? And the other thing is, if they only do one spot instead of two, is that a problem as well? So those are 2 quick questions I had for you.
Okay, so some time back, the drying medium was changed from silica to calcium chloride, and this is a much powerful drying medium. So actually now it's not necessary to dry the sample before you put it into the aluminum envelope. I know that it says 10 minutes drying in the kit, and I think this is a good idea because it will not smear and so on. So 10 minutes drying and then put it into the envelope, it's totally good. It's not a disaster if you don't dry it at all. And when it comes to the 1 or 2 spots, I mean, we can actually do the analysis from a really, really tiny sample. So the robotic system requires an almost filled spot to work. But for those samples that don't have enough blood for the robotic system to work, it's kind of run manually on the side. So it's very rare that we are not able to analyze the sample. But of course, there need to be some blood. Yeah.
Okay, thank you.
So in total, I checked yesterday, it was 535,922 samples processed for Cenzino. And this is quite a lot. We actually have like 20 systems running 24/7 on this now. I think it's one of the largest fatty acids. I would say it's very likely that this is the largest fatty acid facility in the world now. And we have a really automated system. This is some pictures from the lab where you see the robotic system doing the handling of the DBS cards. You can see we have stacks with 20 cards at a time, in a total of 140 cards per robotic system. And there is an arm picking up the card. It's an imaging system that will read the barcode into the database. And actually there is a smart imaging system that will locate the blood on the card. So the blood is not always in the middle of the circle, sometimes out on the side, and this system will then find the blood and punch small pieces of paper into this vial. So you can see we have paper with blood on here. And then we have other robotic system adding liquid, heating, shaking, doing the chemistry. We put the cap on, and then we move this into the chromatographic system, doing the separation of the fatty acids. So we've been practicing this for like 15 years now, and it's really robust, really smooth. And I kind of call it like a copy-paste system. So we have this algorithm that calculates the capacity, and when we pass 80% of the current capacity, we just add another system. And actually the supplier of these systems is in the floor below. So it's very handy to have another system installed. And I would say if you ask the management in Cincino, they will tell you that it's almost flawless now. It's so few errors, it's working very smoothly, it's highly automated, and this is why we are not afraid to say that we are ready for 1 million samples per year, if you can provide it. So I say bring on the samples, it's no problem. So that's almost it. Then I have a few slides which I've only shown once in Munich, like a year ago or so, half a year ago. So I just thought I would give you a sneak peek into some statistics that we are working on based on the 500,000 samples. So this is data from Q1 2020. So it's half a year old. And here you can see a heat map of the world with the sum of EPA and DHA. So in this case, red is bad and green is good. So you can see India is really red, as well as the US. And some countries in Europe like Bulgaria and Romania is also quite red. While the Nordic countries are green, and they have the highest, the best balance, or not the balance, but the sum of omega-3 in the world in the Cincino cohort, while Australia is yellow at the time. So these are the numbers. You can see Australia on the top here has a mean of 4.1 for the sum of EPA and DHA, and it's 1,900 people going into this average. The minimum is 0.5 and the maximum is 13.5. So if we look at this slide, is quite interesting because there are some questions, and this is an example of how these questions can be used. So we kind of filter the data in two groups. One where people have answered that they have not taken any supplements, no Cincino supplements, no other supplements, and it was 228,000 people fitting this criteria. And you can see the mean concentration then was 3.5. Then the second group is where they have answered that they have used one type of Cincino product, and it was 88,000 people that met these criteria. And then you can see the mean changing from 3.5 to 5.8 in these two groups. And in this figure, you can see all the countries divided. So this is the baseline, no supplements, and this is at least one supplement. And you can see that all countries is going up without exception. So I think it's quite clear that this is working. And this is the whole sample. So this is the 228,000 samples, no supplements, and you can see the distribution of the values of EPA plus DHA. And this is the target 8. You can see that almost no one actually meets the criteria, the target of 8. While after taking the Cincino supplement, the average of the group is moved to the right, and a lot more of people actually have a value higher than 8. And then looking at the ratio, the omega-6-3 ratio, this should go down when you take the supplement. Same two groups, 228,000, 88,000, and the mean then goes from 15.2 to 4.9 in these two groups. And you can see for all the countries, you can see they are all going down. So also for this index, the ratio, it also works. And I also subtracted the data from Australia. You can see Australia, no supplements, average is 15, then after supplement, the average is 6.8.
Wow.
So that's it.
That's really good. Now, Thomas, we've got questions firing in left, right and centre, so I'm going to go back and just ask some of these questions. They're excellent questions because a lot of them are what I was going to ask as well. So Gabriella just wanted to verify if we touch the paper while providing the blood sample, will the result be compromised? And I think your slide mentioned that yes, if we touch the blotting paper, it will contaminate, but to what degree, Thomas? Is it a wipeout, or we can't use that test anymore, or do you sort of expect that there's a little bit of contamination going on?
No, we don't expect any contamination, and I think it's quite clearly stated in the instructions that you should not touch the paper. Of course, you can have small amounts of grease on your hands, and then the contamination will not be that bad. But if you kind of recently used hand cream, it will be a disaster for sure. And the ratio is less affected by the contamination than the other parameters. So, for instance, of course, the saturated fatty acid will be grossly affected. But all will be affected since there is kind of a relative analysis. So the C16 will go into the calculation and affect everything. Some in extreme degree and some in a less degree. And the ratio, omega-6 ratio, is less affected.
Oh, that's excellent. I think that's cleared a lot from people's minds. So, folks, I guess the key thing is that when you're getting people to do this test, even though it's a home-based test, it's still important to educate people around that, getting the test done right. I'm a bit fussy. I always go through it with my people. So I don't know how much time people have, but I think I do highly recommend that you educate them. Scott Burgess has asked a couple of really good questions. If the test isn't sent immediately, does it have to be refrigerated?
No. I mean, what is immediately? We recommend to send it the same day or the day after, mostly because if you don't send it the second day, you will probably forget to send it anyway, right? So no delay, I would say. There is no reason to have a delay. But if you know you will not be able to send it within a few days, I would put it in the fridge.
You would put in the fridge. Okay, and how long does it last in the envelope unrefrigerated? Because I know you've got that little preservative in there.
Yeah, so we have like 14 days validated, but we also have data for 20 days, but we don't have data for more than 20 days. So max 20 days for 10 days preferably, but I prefer to send it the same day or the day after. Yeah. Okay,
great. And so those tests that sort of maybe go astray, can we still count on the results, or like what should we do in that regard? If it's taken 4 to 6 weeks to get the results back because it's got held up in the mail or something, what's the best thing to do there?
I would take a new sample.
Right. Okay. Good answer. Thank you. Is there now? Kenny has asked a question. These are great questions, guys. Thank you so much for sending them in. Is there data on test results of children, and what's the average of their results? I think we had a quick conversation around that, but are you really putting the numbers based on adults, or would children be different?
This is all ages that I show you now, but we did look at children below 16 years. And there is some thousand persons below 16 years of age in the cohort. They are not very different from the adults, I can say, but they are slightly different. So I guess this is something that the Ensino management or the scientific board would need to order, kind of extract of this, and have it kind of presented on a conference like this or something else. Yeah.
Yeah, that's great. I think we'll organise a call where we can get into the nitty gritty of the markers of the test. So that would be really, really good. So just coming back to Gabriella, when your UPS touches down, how long would it take for the sample to arrive at Vitas facility? Well, that's a tricky one, Gabriella, I think, because it's going to be determined by the people making the delivery, not so much Vitas. But on average, Thomas, do you know?
31 hours.
31 hours. Okay, thanks, Rob.
From when it leaves my UPS here in Brisbane, it takes 31 to be in Thomas's office.
Okay, fantastic.
There's 2 direct flights a week to Norway from Brisbane, Australia. Okay, that's why we do it twice a week, because it's a direct flight. And within 31 hours, his office has signed my document.
Okay.
So I think this is, to seek a direct flight from somewhere close to the country and have a hub there. This is the way to do it when you experience like 4 or 5 weeks. I think that you should not continue to ship that way. You should try to find another solution using UPS.
And we're doing the same thing with Hong Kong, China as well. Cherie and Thomas, we've got the same setup happening, all going to our Hong Kong office. And Peter's currently shipping 3 times a week out of Hong Kong, and we're not open yet, because we're getting the samples across to get a database of the analysis now, of the Hong Kong people, Chinese people.
Yeah.
Okay, that's great. So just going back to the children, the children's data, would the requirements for children be more than adults generally or less than adults generally? Do you know?
You mean the supplements, the dosing and so on? Or
meaning the how much they require, like for instance, for adults for EPA, it's 4.1. Would children need less than that as an average or more than that?
I don't know. I cannot answer that.
Okay. All right. Well, thank you for at least entertaining that question. So with regard to, we've got a couple more tricky questions which you may or may not be able to answer. I think Scott was sort of wondering why on the silver envelope there's a date, and look, when you can't really write on the silver, he was saying, can that be removed? Like what's the reason for that? Writing on the silver. It's a bit.
Yeah, it's an off-the-shelf type of product. This is not kind of custom made for. So it's the way it's produced.
Okay, but we don't have to fill that in. Is that right?
No, you shouldn't. Well, you could fill in the date, but you don't have to. You're not supposed to. If you were supposed to, it would be in the instructions, right?
Okay.
So yeah.
Great, excellent. So I'm just trying to see if there's.
And can I just have one comment, because some people actually, or a lot of people, write their name on the envelope or someone else, or even birth dates and names and personal numbers and things like that. And we don't want anything of personal information, because this is so important to adhere to the GDPR requirements in Europe. So don't write your name on the envelope. Yeah.
Okay, so that was going to be my question, was around the code, and I think you've really answered it, is that's why it is a coded test, right? Because it needs to be anonymous, because there is a law around this to keep privacy rules, people's privacy there. Is that right?
Yep, that's right.
Okay.
So yeah.
No, I was just going to say that's fantastic. Thank you.
Yeah. So the code is inside the kit, and the kit is sealed. So this is why it's only the person that opens up the kit that will have access to the code. So this is randomized, printed during the printing, and there is no way to know what code is inside what kit. So the person that opens the kit is the only one that knows the code for this blood sample. This is why it's truly anonymous, and then it's not covered by GDPR actually. Okay,
now that's excellent. Now, you may or may not, this may or may not be relevant. Just tell me, I know you'll tell me if it's not, but essentially if somebody's got a pretty serious chronic disease, or there's something going on with their blood, would that impact the testing? Or is this a standard, red blood cell test that won't be impacted by it if somebody's got a blood disorder or a really chronic disease?
I don't think this will affect the analysis at all. So I think the result will be valid even if the person is sick. Yeah. Yeah. Okay. And so I'm just trying to gather all of the questions. Rob, can you see any that I might have missed?
Yeah, sometimes, and I've seen by the questions there, sometimes if people are helping people do the test, because it is a home test, there's always a partner or a friend or somebody or whoever, it may be a nutritionist or whoever, may just say, listen, can you put your phone number on the card as well? Because what we like to do is, and for an example, as a naturopath yourself, okay, Cherie, you would then have the results come back to your email because your patient is getting service. Sometimes partners will do that same thing. They will assist with the reading and the explanation of the data that's being supplied. So I don't see any issue in that, but certainly from the independent side, okay, with the Vitas laboratory, is that they're totally independent. They don't want to see anything or know anything about what's on that dry blood spot. But on the actual customer card like this, I put the date and I normally put the person's name, okay, and and normally it's just their information because I assist them in the reading of it. So but it's a card that goes to Vitas, is where Thomas was referring to, which we don't write any information on that card at all, because that's the independence of the whole thing. And with a little bit of data which has been shared through Thomas, okay, they're just statistics they do to show what's happening with the Omega and the dry blood testing and the age groups of people that are around the world. And now we're testing because we know vegan's going to be a very big part of the Omega world long term. And obviously at Zenzino with the cutting edge technology, it's going to be a big part in the world as we go into that next generation of the vegan product.
Absolutely fantastic. Now, I've got one last question for you, Thomas. I really appreciate your time here. We talked briefly about the 6:3 ratio. I think the two markers that are very commonly understood from all of the common research and literature out there is the Omega 6:3 ratio. Can you tell me where you're getting that from? Is it the World Health Organization? Is that the recommendation that you're following?
Well, I heard Zenzino representatives refer to WHO as a source for this reference value but I really don't have the exact scientific reference for this. Okay. But as I understand it, it's a WHO recommendation, yes.
Okay, and then the Omega-3, we chatted a little bit, and you said that was sourced from the like an Omega-3 index from some research that was done in the US. Is that right?
Yes, there are quite a number of publications on this, maybe 100 actually, in high quality papers where they have studied the risk of cardiac diseases, especially cardiac arrest. And they have found that the risk is substantially lower if you have an omega-3 index above 8. And it's much higher if you have a value below 4. So I think these are pretty well established values. So it should be as close to 8 as possible. I mean, between 6 and 8. And if you're able to, even higher than 8. I think this is quite well established. Yeah. Excellent.
Well, unless, Rob, you can see any other burning questions there, I think that's covered it off from me. It's, wow, this has just been such an incredible talk. Thomas, we're so grateful because obviously I've been running this test for a lot of people now, and it's just given me clarity around how this all works. And it's just really juicy information. I think a lot of people here at the Zenzino family would really appreciate knowing all of this in-depth stuff. I talked to Rob last night about organising a panel of scientists to go into the markers specifically. So guys, get back on that call when we do that one, because we'll deep dive into, the fatty acid cascade and what it means, so we can educate our people. For those that really want that information. Now, just remember, customers don't always want that really in-depth information. It's really people that are understanding the importance and the relevance of this test that really want to deep dive. So, just bear in mind from a practitioner point of view, I know I love the deep science, but I know customers don't always appreciate it. They get a bit confused. So we'll organise that one. I've got Rob already on the case doing that. So, Thomas, thank you so much for your time. Rob, I'm going to hand it back over to you. I've thoroughly enjoyed tonight. Thank you.
Fantastic, guys. And again, thank you, Thomas, your time. 45 minutes of your valued time is just so unbelievably, we just appreciate it so much. And Cherie, thank you so much for doing a spectacular job for us here and adding your expertise in as well. Just to wrap up a couple of those questions that are there, for the guys and you guys in Germany, just think about always talking about express posting, okay? I know people like to send normal mail because it's a few dollars cheaper or a few euro cheaper. We got a COVID pandemic global situation. It's happening around the world, okay? So just get a bit of smarts about yourself and think about it. Get express post happening. It can happen within a few days versus a few weeks, guys, okay? That's why the service level we do here in Australia, we get our test results within 10 days. Most people are 10 to 14 days, unless the person sent the package direct to Oslo by snail mail, okay? If they don't follow the system in Australia, we can only show and give you the envelopes. But, guys, think about that when you're actually talking about the test with the people, your partners, or also your customers. So, guys, wrap up. Thank you very much again, Thomas. Appreciate you very much for doing this. Cherie, as you spoke about, we're going to have Dr. Rizza on the phone, Ole Lander. We got, some incredible speakers coming up in October. Please watch the list. Next week, we got an incredible Chinese naturopath coming along to talk about some incredible technology that she's been working with and how she's implementing this technology into her practices and her consulting that she does here in Brisbane, Australia. So guys, stay tuned for next week. Look forward to always being here on a Tuesday to service this call. Thank you so much, Cherie. Again, Thomas, appreciate you so much, my friend. And when we get back to Oslo, Norway, we'll see you in the clinic. Thanks, buddy.
Yes. Thank you.
Thanks,
Bye-bye.
guys. Bye now.
Bye.
Bye, everybody.
Bye all.
Take care. Thank you guys for joining.